Good afternoon, everyone. I welcome you all to 10th Dr. K.R. Laumas Memorial Oration organized by Department of Reproductive Biology, AIMS, New Delhi. And we have Professor Vasantha Padmanabhan as the orator of the day. To begin the proceeding, I request our Honorable Director to say a few words of welcome, please.
Good afternoon, Professor Talwar, other senior faculty members, ladies and gentlemen, students. It's indeed my pleasure to welcome you all to this 10th Dr. Laumas Memorial Oration, which we've been holding pretty regularly. But this is a challenging time in the era of the pandemic to hold the oration. It's challenging because we're doing it in a sort of hybrid mode where we have some people here, but majority of them are online. It's also an opportunity because it gives one the ability to really reach out to people internationally, and they can, without having to be physically present here, still deliver the oration and be able to educate so many people in a manner which was not possible in the past. So there has been some learning as far as the pandemic is concerned, as far as academic activities are concerned. And I'm really delighted that we have an esteemed speaker for this oration. So I'd like to take this opportunity to welcome her also to the Orange Institute of Medical Sciences. Thank you very much.
Thank you, sir. Now I request Head of the Department of Reproductive Biology to introduce the oration and the speaker.
Good afternoon, respected Director Sir, Professor Talwar Sir, and dear colleagues and students. Today we are here for 10th K.R. Laumas Memorial Oration. Professor K.R. Laumas, Keshuram Laumas, he was PhD as well as MD. He did MD from Mexico and residency in psychiatry from USA. He born in 1933 and died on 1995 in USA. Our department, he was from the inception of the department, initially as a, when our department was as a unit, as a research officer, and when he left this institute, he was HOD of the department. He was in the department from 1963 to 1980. He conducted one international congress, that is 5th International Congress on Hormonal Steroid in 1978, and in the capacity of organizing secretary. Thereafter, he became the president of that International Hormonal Steroid Society, as well as Endocrine Society of India. He was also trained in reproductive endocrinology in NIH as a NIH fellow in Harvard Medical School, as well as State Foundation for Experimental Biology in Massachusetts. He did MD from Juarez, Mexico, and residency from Columbia University Medical Center, New York. This will be the 10th oration. And previous first oration was given by Dr. A.K. Mukhopadhyay from Germany, second by C.P. Puri from Mumbai, then third from C.V. Rao from USA, then Dr. Sujay Guha from Kharagpur, then Sir G.P. Talwar Sir, then Indira Hinduja Padma Shri from Mumbai, Hinduja Hospital, then B.N. Chakrabarti from Calcutta, then Madam Rajlakshmi from Delhi, now she is in MCI. And last year it was given to A.J. Rao. He was from IISc Bangalore. This year award oration will be given by the Professor Vasantha Padmanabhan. Dr. Vasantha Padmanabhan is a Professor Emeritus of Pediatrics, Molecular and Integrative Physiology, Environmental Health Science, Obstetrics and Oncology, and many other few domains. She is PhD from IISc Bangalore in 1970 in cytogenetics. She was a lecturer in Guru Harikishan Public School, New Delhi for a year, then left to USA as a assistant research scientist in Michigan University, Department of Pediatric Endocrinology, and there she became director, pediatric endocrine research and pediatric in Michigan University, then professor pediatrics, obs-gynae, molecular and physiology, and environmental health sciences in Michigan University till 2019. Now she is a professor emeritus in Michigan University. She is editorial board member of various journals, and she published more than 200 publications in high impact factor journals, more than 60 book chapters, more than 350 journal abstracts. Her area of work is translational research on understanding the developmental origin of pubertal and adult reproductive and metabolic disorders, specifically focus on impact on maternal exposure to native steroids, testosterone and estradiol, and environmental endocrine disrupting pollutants, bisphenol A and phthalates, in altering developmental trajectory of fetus and programming adult disease like PCOS, utilizing integrative approach to understand the mechanism by which steroid program reproductive, neuroendocrine, ovarian, and metabolic defects such as PCOS and identify prevention and treatment strategies. So this year oration talk entitled is Programming PCOS Phenotype. It's a very interesting probably. PCOS is a very common disease. Still lot of debates is there. There is no still we are debating on the definition of PCOS. There is no clear-cut definition, and there is no good biomarker except recently as a AMH. And treatment is very pathetic, very poor prognosis, especially those are resistant cases. And it's a totally heterogeneous group of disorders. Now probably Madam Vasantha will enlighten us with this topic, Programming PCOS Phenotype. Good morning, Madam. Now session is 2 yours.
I'm really honored to be the recipient of the K.R. Laumas oration, whose vision has helped create an infrastructure for research in reproduction in AIMS. The topic focus I have been asked to focus on aligns well with this vision to foster research to ameliorate reproductive disorders. Polycystic ovary syndrome is a common, complex, and heterogeneous reproductive disorder affecting up to 20% of reproductive aged women. The diagnostic features include ovulatory dysfunction, hyperandrogenism, and multifollicular or polycystic ovarian morphology. The 1990 NIH meeting emphasized menstrual irregularities and hyperandrogenism as diagnostic criteria. The 2000 Rotterdam Consensus added polycystic ovarian morphology and required presence of 2 out of 3 criteria. The 2010 NIH evidence-based methodology expanded the Rotterdam criteria with subclassification into subphenotypes. The PCOS subtypes reflect different combinations of the 3 diagnostic criteria. Type A with all 3 PCOS criteria, type B, hyperandrogenism and ovulatory dysfunction, type C, hyperandrogenism and polycystic ovaries, and type D with ovulatory dysfunction and polycystic ovarian morphology. PCOS is associated with several reproductive consequences. The ovulatory dysfunction may manifest in irregular cycles and abnormal uterine bleeding, anovulatory infertility, endometrial hyperplasia, and endometrial cancer. There are increased risks of preeclampsia, gestational diabetes, prematurity, and both large and small for gestational age babies. A large percentage of women with PCOS manifest metabolic dysfunctions that include obesity, insulin resistance, dyslipidemia, and impaired glucose tolerance, type 2 diabetes, and hepatic disorders. They're also at higher risk for cardiovascular disease. Evidence point to both mood and sleep disturbances and reduced quality of life. They're more likely to have obstructive sleep apnea and at a high risk of depression. So in addition to the 3 diagnostic criteria highlighted in blue here, PCOS women also manifest high LH, reduced sensitivity to steroid feedback, Ovarian dysfunction manifested as increased follicular recruitment, increased follicular persistence, and they also manifest insulin resistance. Furthermore, obesity amplifies the phenotype. So what are the basic pathophysiologic mechanisms contributing the reproductive and metabolic defects associated with PCOS? The hypothalamus pituitary ovarian axis, shown on the left, is central for the regulation of the reproductive cyclicity. GnRH produced in the hypothalamus drives pituitary production and release of FSH and LH, which in turn act on the ovary to regulate follicular growth and steroidogenesis. Precise coordination of components of this axis is essential to maintain cyclic reproductive function. Several feedback loops exist to maintain this homeostasis. In PCOS, shown on the right, the HPO axis is disrupted at each of the three levels, thus contributing to the reproductive defects we observe. Reduced steroid feedback, leading to increased GnRH drive, increasing pituitary LH secretion that stimulates ovarian theca cells to produce testosterone, excess testosterone, contributing to hyperandrogenic status. At the ovarian level, primordial follicles, upon getting activated, transition through primary preantral, antral, and preovulatory follicular stages. During each of these steps, there is attrition of follicles as a subset of them undergo atresia. In PCOS women, shown in the bottom, there are disruptions at multiple levels. First, there is increased recruitment leading to increased number of growing follicles. In addition, there is reduced atresia leading to follicular growth arrest. The increase in recruitment coupled with follicular persistence contribute to the multifollicular appearance, one of the diagnostic features of PCOS. In the metabolic side, nearly 70% of women with PCOS manifest insulin resistance. Women with PCOS are characterized by systemic dyslipidemia, adipocyte defects, and are at increased risk of ectopic lipid accumulation at peripheral sites, leading to lipotoxicity. These metabolic defects associated with PCOS contribute to the insulin resistant state and compensatory hyperinsulinemia. Hyperinsulinemia in turn influence the HPO axis to increase LH secretion and increase adrenal androgen production and reduce liver production of sex hormone binding globulin, leading to increased free testosterone in circulation. All these metabolic defects in turn contribute to hyperandrogenic state, one of the key diagnostic characteristics of PCOS. Then the neuroendocrine, ovarian, and metabolic defects associated with PCOS suggest that the outcome is a self-perpetuating vicious cycle that aids in continued maintenance of the disease states. GWAS studies have linked multiple genes with PCOS, which together only account for about less than 10% of the prevalence rate of PCOS. There's increased recognition of gene-environment interaction as the basis of development of PCOS. Based on this premise, genetic susceptibility to PCOS would be subject to modification by the prevailing environment. Insults during the critical periods of development can bring about organizational changes in the developing organs. Use of novel animal models of PCOS have the potential to not only contribute to our understanding of pathophysiology, but also to the developed interventions for prevention and treatment of PCOS. We have developed the sheep model of PCOS phenotype. Historically, sheep have been valuable in the study of maternal fetal medicine. They are precocial species, just like human, and have similar developmental timeline like human. Using pregnant sheep, our laboratory has developed a sheep model of PCOS. This involves injecting testosterone to the pregnant mother and then studying the female offspring, which is our test subject. So why do we study sheep? there is a lot of similarity between sheep and human. Shown here are the hypothalamic, ovarian, and pancreatic development in utero comparison between sheep on the top and human in the bottom in each panel. So as you can see, the complete GnRH LH system, the neuroendocrine subsystem develops in utero in sheep, similar to that of human. Same thing happens at the level of the ovary, where a complete follicular differentiation occurs in utero in sheep, just as is the case in human. At the pancreatic level, sheep also have all the islet formation completed in utero, which is very similar also in human. So the red bar here shows the time period when we are treating the sheep, pregnant sheep with testosterone. Sheep have 147 gestational days, and we treat them from day 30 to 90, which encompasses a sexual dimorphic window when normally a male fetus sees an increase in testosterone. So this really shows the treatment paradigm. So you're treating from day 30 to 90 of 147 gestational days. For reference, in future, all the controls are shown in orange and the T treatment in yellow. So this shows maternal and fetal levels of testosterone achieved in relation to our injection of testosterone. So at the maternal level, what you see is increase in testosterone because we are administering it, and these are in the male levels. And at the same time, the increase in testosterone causes the mother to be hyperinsulinemic. So mother is both hyperandrogenic and hyperinsulinemic. At the fetal level, if you look at the female fetuses, the increase in testosterone leads to about 0.5 nanogram in the female fetus. In addition, the female fetus also sees an increase in estradiol because testosterone can be converted to estradiol, or this may be coming from the fetal gonad. In comparison, if you look at the male fetus, the male controls normally reach about 0.5 nanogram of testosterone. So what you're giving is very physiological and similar to what a normal male fetus sees. The next slide shows what cord levels of testosterone in human fetuses. This happened to be a study we did in early 1991. Serendipitously, we were using cord blood from human fetuses, and you can see about 40% of the female fetuses also have levels of testosterone in the male range. This doesn't mean 40% of these fetuses are going to develop PCOS, and there's a genetic susceptibility component to this all. This study shows cyclic patterns of progesterone, which are in the sheep, that's control on the top. Those receive 60 to 90 days of treatment of testosterone, and those that span the 30 to 90 day window. As you can see, controls are cycling normally. Sheep are seasonal breeders. So there is a period of an estrus period where there is no cycle, and then they resume cycle again during the breeding season. So they're repetitive cycle, 100% of them cycle throughout. Those that receive treatment from 60 to 90 cycle the first year and also cycle, but they showed some aberrations in cyclicity, so there was some subtle differences. The 30 to 90 animals, which is the model we predominantly use, began puberty early, but they cycled early on, but they stopped cycling and became anovulatory. Some animals do show irregular cycles, but majority of them showing anovulatory cycles. I mentioned PCOS women have LH excess. This shows 3 sheep on the top, cyclic, pulsatile LH secretion on the top, and the PCO sheep in the bottom. As you can see, the T-treated sheep have higher LH and high amplitude LH pulses. So the LH excess is another feature of women with PCOS. The next slide shows the responsiveness of the pituitary to GnRH. Here we shut down the endogenous GnRH with steroids and administer exogenous GnRH. And you can see the pituitary is responding with each pulse of LH to a bolus of GnRH. In the bottom of the T-treated, you get a higher responsiveness to the GnRH injection. In the right, it shows the increase in amplitude of LH in response to GnRH. So not only there is increased drive to the pituitary, but the pituitary is highly responsive to GnRH, a feature also seen in women with PCOS. So at the ovarian level, it shows on the left of the ovarian weight, they have higher weight in the T-treated animals. The antral follicular numbers are increased. The ovarian reserve doesn't change, but the follicular, large follicles are increased. If you look at the ovarian morphology on the right, controls are showing follicles on various stages of development. That's what normally one would see. But the bottom panel shows prenatal T, where follicles are getting arrested at the antral follicular stage, which is a characteristic feature of polycystic ovarian morphology, where you see a large number of small antral follicles accumulating at the arrested stage. So these are daily ultrasonography. We track follicular development in sheep. We track each follicle by daily ultrasonography. And then we have done up to 21 days of tracking. These are control animals, just expanded to show how follicles are emerging and regressing over time. And the next slide shows follicles in the T-treated animals. Here you can see follicles are emerging and sort of hanging around. They're not dying out, they're not regressing. So this is what contributes to some of the multifollicular ovarian morphology. Estrus, when we looked at fertility status, when we introduce a RAM, Rams prefer controls over the T. They avoid the prenatal T-treated sheeps. When we remove the control, they do breed the T-treated sheeps. What you can see is only about 40% of the T-treated sheep got pregnant. So fertility was heavily compromised. On the right-hand side, you see the control herd, where you see over 90% of the sheep getting pregnant. So I want to point out these are 60 to 90 treated sheep, which are little compromised in terms of only mildly compromised compared to the 30 to 90, which are even more worse than this model. When you looked at the metabolic side, these shows responsiveness to a glucose challenge, glucose on top, insulin on the bottom. You can see animals are weight matched. They're not obese. And the testosterone-treated animals show a heightened response of insulin. And this on the right, shown on the bar graph on the area under the curve, and the insulin sensitivity index, which is the opposite of insulin resistance, is reduced. So these animals are clearly becoming insulin resistant. 70% of the women with PCOS do manifest insulin resistance, as I mentioned. So then we looked at the hypertension in terms of blood pressure monitoring. We did remote blood pressure monitoring using telemetry devices. And you can see the prenatal T-treated sheep, when they become adult, they're hypertensive. These are mean systolic arterial pressure. And the next one shows mean diastolic pressure. And you can see teat animals do have a heightened blood pressure. So the outcome is at all levels, there is a vicious self-perpetuating cycle. At the neuroendocrine level, there is advancement of neuroendocrine puberty, reduced estrogen negative feedback and positive feedback, reduced progesterone negative feedback, increased pituitary sensitivity to GnRH. All these features contribute to the LH excess that we see in women with PCOS. At the ovarian size, there is no change in ovarian reserve, but there is increased follicular recruitment, increased follicular persistence, multifollicular ovarian morphology, that's a characteristic feature of women with PCOS, as well as corpus luteum defects. At the cardiometabolic side, you do have reduced insulin sensitivity, topic I didn't have time to go into. They do have alteration with visceral adiposity. They do have increase in total free fatty acids condition leading to dyslipidemia. There is also increase in blood pressures. So all these defects at the T3 sites, the neuroendocrine, ovarian, and the cardiometabolic sites, lead to this vicious cycle, resulting in outcomes which leads to pubertal advancement, oligoanovulation, reduced mating success, reduced fertility, insulin resistance, and hypertension. So this slide summarizes the features of prenatal T-treated sheep, the model that we have created, versus the PCO, women with PCOS. You can see pretty much we have recreated everything that a woman sees, a PCOS woman sees in sheep. They're anovulatory, they have LHXs, they've reduced sensitivity to feedback, they do have the polycystic ovarian morphology, follicular recruitment is affected, they become insulin resistant. They do show altered behavior, which I didn't go into. These sheep do show more aggressive behavior compared to controls, and these animals are also hypertensive. Furthermore, obesity amplifies the phenotype. So when we think about organizational versus activational effects, there is a critical window. In our case, it was 30 to 90 days, where any insult can lead to organizational changes, which can ultimately lead to permanent changes. And this is where you need to intervene if you need to undergo any kind of prevention strategy. At the bottom is the activational effect. Activational effect merely means that you do need the insult present to see the symptomology or pathology, but when you release them from that symptom, you can release them from the symptoms too. So I think this is where the PCO field right now is, where we are focusing on the treatment to ameliorate some of the effects, but the permanent changes are still persist. So I think we need to be thinking as we move forward with PCOS in terms of prevention strategies. So the two features of women with PCOS are the hyperandrogenism and the hyperinsulinemia. This is where most of the targeted treatments are directed to. So I mentioned excess weight gain increases severity of reproductive disruptions. And these we actually tested. We generated the control and prenatal T-treated sheep. The crossbar from top to bottom shows where we put half of those animals of higher, overfed them. A batch of control animals and batch of prenatal T-treated animals were overfed. So we increased body mass by about 30%. So you have control T-treated animals, T-treated animals which were made obese, and control animals which were made obese. And you can see they're pretty well controlled. We are weighing them periodically and adjusting. So ultimately, you do lead to weight is not an issue between control and treatment. So what happens in terms of cyclicity? These are 4 panels. These shows luteal progesterone cycles in terms of anovulatory responses. You can see controls are all mounting a luteal progesterone increase. They're all showing progesterogenic cycles, showing they are ovulatory. When you come to the next column, which is the control obese, you're seeing certain anomalies in terms of the progesterone pattern. So there is some cyclic disruption. The third panel shown in yellow, which is the T animals, you can see many of them are not cycling, or they have inadequate progesterone levels or insufficient, so they're having major effects. When you come to the last column, where the T animals were made obese, you can see pretty much most of them became anovulatory. There was one animal that did cycle. This brings up against the gene-environment interaction, because not everybody is susceptible to insult the same way. This one shows the impact of postnatal obesity on insulin sensitivity. And you can see, again, on the left are shown, on the bar graphs are shown the weights of these animals. You can see control T are pretty closely matched. Control obese and T obese are pretty closely matched. When you looked at their insulin responses shown in the bottom panel on the right, you can see control animals are responding to glucose challenge with increased insulin. The T animals are showing an increased response to the same glucose challenge. Obese controls also show a heightened insulin response. And the topmost one, which is the dotted line with closed yellow circles, shows the obese T, which shows that these have a much more magnified insulin hyperinsulinemia, and they tend to be more likely to be more insulin resistant than the other animals. So the first 5 years of this grant, we had a program grant to develop the model, and then we started focusing on intervention. So we were focusing both on prevention and treatment. And these shows the groups of animals we generated. We generated the control and prenatal T-treated animals. And we generated a group of animals where we gave prenatal T-treated animals with flutamide, which is an androgen antagonist. And then we also generated a group of animals we were giving testosterone as well as rosiglitazone to modulate their insulin sensitivity. So you're hitting them from both angles. I mentioned the mothers become hyperandrogenic and hyperinsulinemic. So we are controlling for those two, negating those two actions to see which is the program agent. So these are the prevention arm, and in the very bottommost, we show the treatment arm, where we generate the T animals, and towards the end, we give them after the animals are generated and the phenotype is established, we treat them with the androgen antagonist or rosiglitazone. So these show some of the results. I wouldn't have time to go into a lot of results. There's a whole 10 years worth of work here. And I'm only summarizing the highlights. And you can see on the top, the controls animals are mounting a normal LH surge. And then you come to the T animal shown in the middle. There is one animal that's showing an LH surge, but much delayed onset of the surge. So these lead to chromosomal anomalies in terms of reproductive phenotypes. And the other two animals did not even surge. So when we gave them flutamide, which is the androgen antagonist, you can see we restored the surges, but we did not alter the timing. So there was still a delay, showing that a component of that is reversed, but not all of it. So then here, looking at the luteinized follicles, and you can see T animals leads to a lot of luteinized follicles. Androgen antagonist reverses some of this, and rosiglitazone only partially reverses it. When we look at the insulin resistant arm, when the reproductive side was beneficial by androgen antagonist treatment, the reproductive side, the androgen antagonist treatment did not help prevent the development of hyperinsulinemia. On the rightmost, you can see rosiglitazone treated animals did improve development of hyperinsulinemia. So you're really dissecting out the reproductive and metabolic consequences of prenatal TXs, and it tells you that maybe both are needed. So this summarizes all the work that we have done, just to give you a sense of what's going on. So there is a lot of things that are involved, and you can see I'm not going to go into details because of the timing. And I want to point out almost all the reproductive features. If anything, the androgen negation seems to help to ameliorate. All the yeses says that they do help, and in terms of others, it doesn't. So you can look at the ovarian transcriptome, follicular development. You can see all these aspects are benefiting from androgen negation. And on the metabolic side, most of them are negated by the insulin sensitizer treatment. So normalization may require targeting both the androgenic and metabolic pathways. And in addition, we also need to think about the importance of estrogenic pathways, because testosterone is getting converted to estrogen, and therefore there is potential for estrogen also acting on this cascade. So the adult phenotype, what we develop, is a function of what insults they achieve during development, during their fetal life, as well as what activational signals that are available to establish your final phenotype. So in case of these, there is a genetic susceptibility that organizes the system, but environment and lifestyle factors through maternal milieu and epigenetic changes can influence this and induce some alter the susceptibility of these individuals. This in turn programs endocrine alterations, which itself sets up the activational effects, which could be ameliorated by treatment strategies. So I think, we need to think about this like a two-hit stage where you have a first hit which organizes and a second hit which could amplify it. And one of the concern is some of these program phenotypes gets carried on to the next generation. So animal models have been very helpful in terms of ensuring human health. So in terms of future, with many scientific advances in PCOS, there are abundant possibilities for new preventive, diagnostic, and treatment strategies for women with PCOS. Careful phenotyping of women with various PCOS subtypes may help in personalized and targeted treatment. Phenotyping and following up of children longitudinally will allow early prevention and intervention. There is also a male PCO phenotype, and those have not been as well studied, and these do have sperm compromise. And so these may help clarify male phenotype and determine risks that need to be addressed as well. And even more so, and finally, assessing the importance of impact of environmental exposures, such as stress, food, chemicals, and air pollution, are important to understand. So there is a clear interaction between gene and environment, which we need to understand so we can lead to public health strategy to reduce risk of PCOS. So I'd like to thank all the students and postdocs and undergraduate students. I mean, many of them I didn't have space to even list, who participated. These are long-term studies, which goes on over, we program them, and the adult animal we study 3 years later. So you can see the time span of all the studies that undergo, where all the tissue-specific studies get done. We received farm support from Doug and Gary, who are amazing herdsmen, and really we couldn't have done these studies with them. At times we have bred about 400 animals. Most of all, I like to thank the National Institute of Health, who provided the funding, several $1 million, to get some of these work done. So we have a model where we can develop intervention strategies. Thank you.
Thank you, ma'am. Now it is time to present the award. May I request our Honorable Director to present the award?
डाइनिंग हॉल में सर टी करेंगे डाइनिंग हॉल.
I'm honored. Thank you.
Thank you, sir. Now I request Professor Rajiv Kumar, Associate Dean Academic, to deliver vote of thanks.
Good evening, Professor Padmanabhan, the orator for today, the director, Professor Andeep Guleria, head of the department, members of the faculty, students, staff, guests who've joined in today. Thank you very much for coming in today to listen to this oration. First of all, a thanks to Dr. Padmanabhan for accepting our invitation to deliver the oration today. We understand it's difficult, it's very early morning, and I'm sure it's pretty cold at the moment, despite it being April here. So thank you very much for coming up early morning to deliver this talk. It's a late evening for us, but early morning for you. And I also like to thank all of you in the Department of Reproductive Medicine who've gathered together today to attend this oration and keep up this spirit of having an academic discussion and an oration alive despite the difficult times that we seem to be going through all across the world. We're seeing a resurgence. We don't know. We had hoped we would get back to normal by this time this year, but it seems that we are repeating the cycle of what we saw exactly on the same week last year. So we don't know when we'll get back to normal, but thank you again for organizing this, and to everybody who's online to listen to this oration and to be a part of this academic session. Thank you very much.